Are there any other announcements?
No.
All right, I'll turn it over to Eileen, who will introduce today's speaker.
I'm starting to need a seminar to list all the seminars. Before I introduce today's speaker, I want to introduce a visitor, one visitor who hasn't apparently been introduced to the group, and that's Veronica Carici, who's back here in the corner. You want to raise your hand, Vero? She's here visiting Juliana de Abreu. Veronica's from Uruguay, did her PhD in Santiago de Chile, and is now back in Uruguay and works on social mammal behavior. So she's here. Elfgar's not here. Alfred, oh, you're there. Do you want to introduce your student who's visiting as well?
Okay. Hi. I'm from Uruguay. I worked in Chile for many years, and here is Ertego, that is one of my students. That we have Ertego with Juliana, and Ertego is interested in malaria. So here we work with genomics. So this is collaboration with Raul. So I am learning as well.
Thanks. So Vero's here until Friday, and I don't know how long. How long are you here?
How long what?
How long? You're here till Friday.
Yes.
Yeah,
I'm
okay.
leaving on Friday.
Okay. So, but you can talk to her quickly if you'd like to. We probably should get down to business here, right? You get a 10-minute talk after all this. So it's my pleasure to introduce today's speaker, Jen Smith, who's currently an associate professor at Mills College down in Oakland. And we were trying to recollect, we didn't get to the date of how long ago we first met. We got to, it was at a Mammal Society meeting, and I remember you were a graduate student at the time. So probably last year, right? We've known each other a while. So Jen is from Maine and did her undergraduate work at Colby College in Maine. She got her degree there in 1998. She then ended up working for a PhD at Michigan State University with Kay Holtkamp as part of the famous hyena project that Kay has run for many years, both in Africa, used to use the captive hyenas here on campus, and various other things. Jen completed her degree at Michigan State in 2009, did a postdoc down at UCLA, and then landed her current position at Mills in 2012. She's a phenomenal teacher. She's won multiple teaching awards, keeps an active research program going at Mills, even though Mills is a little bit more teaching focused than perhaps we are. And today she's somehow going to pull together themes related to social behavior, cognition in hyenas, and then these obviously superior animals here, California ground squirrels, that Jen currently has an ongoing active field program here locally in Briones with the California ground squirrels, and she'll tell us a little bit about that. I think just last thing to mention, I'm supposed to mention some fun facts here. So as part of her doctoral research, Jen spent a year living in a tent in the Masai Mara, where not only did she observe hyenas, but she had interactions with other local wildlife, apparently often involving peanut butter and ownership disputes over peanut butter. You asked her about that.
With the squirrels.
That's true. Some things don't change. And as I mentioned, you can sometimes run into her out at Briones. I have. I don't know if you guys knew it, but you pass me when I'm biking in your vans quite frequently. And so you see a bunch of people working with ground squirrels out in Briones. It's probably Jen and her group. Just to conclude, she's very active in promoting STEM education for women, and would be happy probably to talk with people about that later today too. I've said enough. Thanks, Jen. Take it away.
Thank you, Eileen. And I just want to thank everyone for coming out today. And I'm often teaching at this time slot, so it's I've been wanting to come over and join you for this, and it's a great pleasure to be able to come over and do so today. And as Eileen mentioned, I will be tying together some of my favorite animals, spotted hyenas, all the way to ground squirrels. And the major theme will be thinking about factors that promote individuals to interact. And as many of you know, many different species of animals will aggregate when doing so is beneficial, forming loose aggregations of birds flocking and fish schooling. But most of my work actually focuses on individuals that have repeated social interactions. And what drives me and the questions that I'm most interested in is trying to understand what promotes social evolution and what are the rules that give rise to social structures that we observe. And within these variety of different social group living animals, I'm really interested in, although I don't study capuchins, I think they give a nice example of the types of questions that I'm interested in. And these have to do with how social rules governing decision-making, behavioral responses vary with ecological context. So when individuals that know each other move through scenarios such as feeding together, forming a coalition, or simply resting, how do the rules that promote behavioral decisions, how consistent are they? How are individuals looking on a continuum of being highly sociable or less sociable? And different contexts that these group living species occupy are things that include individuals coming together and defending their offspring from predators, or maybe detecting predators earlier on, or maybe actually gaining access to food that they wouldn't otherwise have an opportunity to eat, such as these wolves joining forces to grab a prey item that they wouldn't be able to do on their own. Of course, there are many benefits of group living, but in addition, there are a number of costs, and Eileen and I, along with Lauren Hayes, have a book chapter thinking about these costs and benefits of sociality, and some of these are conflicts that emerge over limited resources. These include food or mates, as well as potential spread of parasites, either ectoparasites on the skin, such as these ticks and fleas here, or endoparasites that might occur internally. And so although there are a number of benefits for animals living in social groups, there are these potential costs that individuals face. And these are the types of contexts that I'm interested in, and the general theme that I will be approaching through different questions today. The 3 lines of questions that I will be addressing are: first, which factors control or contribute to social dynamics within spotted hyena societies? So how do they change over time, and how does prey abundance influence decisions that they make? And then I will switch over to this local system on California ground squirrels, and going a little away from the theme on social networks, I'll tell you about some of our more recent work on risk-taking behavior, and how this might vary with the ecological context. And finally, return back to the theme of social networks and discuss and inquire to what extent are individuals, when they are interacting below ground, having consistent social network traits compared to how these social networks emerge above ground in these spaces where interactions are relatively free and open. So this is where I'm going. I will start off with hyenas, and I think particularly for this crowd, might be fun to dabble in these a little bit. As many of you know, these are large carnivores. They live in female-dominated societies, and the work that I will talk about is in collaboration with Kay Holkamp, who started the Mara Hyena Project, along with Laura Smale, also not a stranger to this audience, back in 1988. And so we've been using these long-term data to ask a number of questions. And these spotted hyenas live in groups that can be comprised of up to 130 or so individuals. And their lives are typified by large-scale cooperation, but also competition. You can see these two are fighting over food once they've acquired it cooperatively. And these societies are structured by a linear dominance hierarchy, and their maternal groups within the overall society actually give rise to this structure. And the way it works is that we have multiple matriline's here of family members, so individuals in the alpha matriline, or the highest ranking within the overall social group. And we have the adult female here, the circle, her daughter and her granddaughter, and so forth. And down this lineage, individuals inherit the ranks of their mothers. So because of this, we're able to actually rank based on the outcomes of agonistic interactions, them in a linear dominance hierarchy, and bracket shoulder is socially dominant to Murphy, to Seinfeld, her daughters, and this goes all the way down the line. This is true for phylogenetic, that's another line of work, phylopatric animals such as the females that remain in their group, as well as individuals that are born there. And we have multiple individuals in these multiple family groups that all belong to this overall clan or social group of hyenas. For males to reproduce, like most good mammals, they actually disperse and leave and go into another clan. And when immigrant males come in, they queue for social rank, and they're all the way at the bottom of the dominance hierarchy. So this gives rise to a group that has genetic relatives that are closely related within a matriline. It also gives rise to male genetic diversity that comes in as they join the group. So on average, there's very low relatedness overall. So very complicated types of social structure, and despite this low level of relatedness overall, these hyenas will cooperate in large-scale warfare. So this is a little bit of a messy video, but it's one of my favorites, showing hyenas from one social group or clan on this side, and then on the other side of the group, you'll see other hyenas from another clan. They're joining forces and they're meeting at a territorial boundary, and we call this a clan war. So I'll show you this little video. Let's see. So individuals are coming in, they're joining forces from one side, and then others are coming in from the other side.
Okay, here we've got because the hyenas rushing at our guys, and they're rushing back.
Yeah, and so a lot of my dissertation work looked at this large-scale cooperation and the rules that were governing ways that individuals are choosing to form coalitions or not. And adult females are typically at the front lines of these battles and are really leading this collective behavior. I've been thinking a lot more about leadership. I'm not going to talk about that today, but if you're interested, heading to Germany at the end of the month to think about it with respect to lemurs and all sorts of other animals. What I am going to talk about with respect to social networks is the fluidity of these hyena societies. So a large number of individuals. If we follow an adult animal, they are spending about a third of the day actually by themselves in a subgroup of one. So they're actually in these really, really fluid social groups where the subgroup size is changing every half hour or so. So they can be in a subgroup of one, in pairs, they can be in subgroups of various sizes. And the ecology determines how they form these groups, as well as social preferences with respect to kinship and social rank. Again, as part of my dissertation work, I followed these spotted hyenas with radio collars. There are two consecutive follows, follow A and follow B on the same individual hyena in the evening, the daylight hours, and again in the evening hours. And what I want you to notice here is that the individuals, if they're alone, they're in a subgroup of one, and they vary in the number of subgroup sizes that they encounter, and these are changing constantly over time. They're changing within a day, but they're also changing between these 2 days. And so this gives rise to social networks where individuals are meeting up with and are actively selecting who to associate with and when. And when they meet up, they will often engage in these greetings, and I'll show you a video. This animal here is an adult female with another adult female. When she arrives at this group, she often will initiate a greeting, and it looks like this. These hyenas are incredibly interesting in their morphology. They have fully erectile pseudopenises. If you look carefully, you can actually see this adult female's phallus has been torn, so she's given birth. This is kind of like animals at the dog park that are coming head to tail, sniffing each other, and some of my work showed that this actually promotes some of that large-scale cooperation, clan wars, two groups coming to battle against each other, joining forces in that way, as well as coalitions within the group overall. And what I want to focus on here is the way that kinship is influencing social network structure. So there are multiple matrilines within the hyena social group, And all of these individuals interact to some extent over time. In this social network, there are individuals belonging to this alpha matriline. The larger circles represent adult females belonging to that alpha matriline, and the smaller ones are subadults. What you can see here, hopefully you notice right away, is that the kin relationships are most tightly corresponding to one another. So what I mean by that is that the thickness of these lines, each one of these lines represents the extent to which individuals are walking up and are associating in space with each other. They're making active decisions to associate with some individuals over others, but all of the members in the clan are linked together by these connections to some extent. And just for a little teaser here, the question that I will ask today is basically, how does variation in prey abundance influence patterns of sociality? So we found that kin prefer to associate with one another over non-kin, but prey vary in their abundance in the Masai Mara. So most of the time of the year we have resident ungulates that are in the area, and then there's a superabundance of prey that come in as the rains come. There are zebra and wildebeest that come in a large number, and the question that we addressed here is, to what extent does the social network vary in its structure based on this large number of prey coming in, the superabundance of prey? How does that influence the kin structure and the overall network structure? And again, these data are from this long-term study, so there are multiple years that we have been looking at this, but I'll just show you several slices of 4-month periods that we included in this overall study. Here there are individuals that are interacting with each other during a low prey period. And what you can see here is that the lines are relatively thin. That means the social connections overall are relatively sparse, but there's still this preference for kin over non-kin with respect to associations. As the migratory prey join the residents, we have a superabundance of this food everywhere. This relaxes feeding competition, and what we see is that the overall density of the social network is higher, and the strength of these individual connections is also higher. If we then go to the next period, you see that the strength of these connections decreases again as the food goes away, and competition is more important, and it actually disrupts patterns in this network. Importantly, kin are always associating most often with their genetic relatives. And although it might seem commonplace now to think about these social networks in these dynamic ways, when we first started thinking about this, a number of us came together and we wrote this review for thinking about ways of advancing the field of social networks. Rather than just thinking about one static network and determining everything that's going on from that one time slice, we really thought that this work and other bodies of work related to it could help us to think about the ways that these are changing over time and social relationships are varying with ecological contexts. So there's a little snippet about that. So for this part of the talk, I've shown that resources constrain social network dynamics, but these kin-biased social ties are resilient to ecological perturbations having to do with how much food is available. Okay. So now switching to California ground squirrels, and to the remainder of the talk I'll focus on 2 different aspects of their biology. And the first will be risk-taking and whether this varies with ecological context. And then second, returning to these social networks with California ground squirrels. So this work, as Eileen mentioned, is part of a long-term study, becoming a long-term study. Hope that it will also go on for 30 or so years, started in 2013 out at Briones. And we have been live trapping and marking individual squirrels. We give them marks such as road here and mill, so if you go out to Briones, you might notice these strange marks, and that allows us with our binoculars to identify them quite readily above ground as they are going about their business. And we study them at 2 different colonies that are close to each other. they vary in the level of disturbance by us, by humans, both off-leash dogs that we bring with us and just humans coming in and out of the areas. So this paradise colony is relatively low disturbance, and if we measure the glucocorticoid or stress hormone metabolites from these animals, we actually find that they are significantly lower in this one area compared to the second colony, which has a lot higher levels of disturbance. So California ground squirrels, as a lot of you probably know, are locally abundant, and they are facultatively social, so they allow for us to ask questions about the degree to which animals choose to be social or to spend time by themselves. They're also very important for many predators will prey upon them, and these predators include rattlesnakes, such as this rattlesnake that's here. You can see our marked squirrels, this is pie here, and volcano will be over here, and so they do this cooperative tail flagging behavior, and they are dealing with predation is very much a part of their everyday experience. These are very obvious, they're above ground, it's easy for us to observe these types of behaviors. So they're clearly preyed on by snakes, although these two are still with us and still carrying on. But birds of prey as well as coyotes will often take these squirrels. And what we've been thinking about recently with Chelsea Ortiz and Erica Pendleton, they're both undergraduates. I have an undergraduate-only lab at Mills College. And so this work is a culmination of their 2 undergraduate theses. And we've been asking, to what extent do these squirrels deal with a trade-off between foraging and looking or vigilance during foraging behavior? So we do focal animal surveys. We identify an individual squirrel. This is based on 5 years of data and roughly 300 focal animal surveys where we observe them foraging in 2 different positions or looking out for predators. And so we're interested in what are the factors that are contributing to this trade-off. And what we find, is these are really classic behavioral ecology kinds of questions, is that these squirrels can forage by themselves when they're alone, or they can actually forage in groups of 2 or more individuals being close to them. And with respect to the rate or how often they are looking for probably predators or other things that are at risk, the rate of vigilance decreases with the group size that they are in. And this is true for both juveniles, like this little one, or adults, this one above. In addition to the group size influencing how often they look, there's another aspect of vigilance which has to do with the proportion of time that they're allocating to vigilance or foraging. And what we find here is actually an age difference in the way that they respond to conspecifics being near them. So here you can see for juveniles, that being alone or with conspecifics, they actually are so focused on eating, they actually don't change the proportion of time that they are allocating to vigilance. However, for adults, we found a significant difference between adults allocating more time to vigilance when they're alone, whereas they reduce their amount of vigilance when they are in a group. And these factors are consistent if we look at it on a group size as a continuous variable as well. There are a number of microhabitat features, so the ecology also contributes to these 2 different aspects of vigilance behavior. And the microhabitat features actually influence different aspects of their vigilance. So if we look at the rate of vigilance, we see the highest rates of vigilance when they are foraging on low inclines. So what that means is they're on an incline that's relatively flat to the horizon. And this is compared to when they're on these higher inclines. They reduce, so they're on a higher incline of greater than 10 degrees relative to the horizon, but they actually reduce the rate at which they look for predators. They feel safer is our inference there. However, there's a second aspect of vigilance that is influenced by another microhabitat feature, and this is how tall the vegetation is. So they actually allocate more time to vigilance when they're in low vegetation. So these are circumstances where vegetative cover doesn't cover the squirrel if they're foraging with all 4 paws on the ground. and they're more likely in our experience to encounter things like that rattlesnake that I just showed you in the video, as well as other predators, aerial predators. And so foraging and this exposed type of microhabitat seems to increase the allocation of time overall of being vigilant. What we're doing more recently, and this work is sort of still in progress, we are starting to do some more experimental types of tests to get at risk-taking within these animals. And this is an animation that Erica put together. Chelsea Ortiz is now starting her first year at UC Davis and has an NSF for her PhD there, and she's continuing to work in my lab on California ground squirrels. And so she's continuing with this work and thinking about flight initiation distance as a measure. As an individual here approaches a squirrel of a known identity, we will put down a flag when we start. We approach this squirrel, and then the squirrel runs away. And when they do, we put a flag where they originally were and where we stopped and they flew away. This is originally thought of for birds, but they, squirrels kind of flee. They can fly. And we also measure the starting distance. And this measure gives us insights into how fearful the animals are, how close we can get to them. If we're able to get relatively close, then these individuals are fairly bold and are less fearful of us. A longer flight initiation distance instead suggests that they are more fearful of being approached by humans. And we looked at a number of different factors in these tests. I'm showing you data that are for individuals that we approached on multiple occasions. Each one of these bars represents a box plot for each individual, and I have individuals that are ranked from least fearful, so really bold ones, all the way to most fearful on the other end. So the individuals that run away when we get, and we're not even very close. And what we find is that the individual identity within these mixed effect models is crucial. So it explains a large amount of the variation in these groups. Some individuals are consistently bold, others are consistently fearful. And what we're starting to think about with these data are why, okay? And what we've been noticing in the data, and we're starting to develop this in more detail, is that within the crow colony, that's where these tests were done, in the more highly disturbed one, there's a outhouse that's here, or picnic tables, people can come along and have a lunch there, and so forth. On this end, and this is really a high disturbance of humans and off-leash dogs. We record all this, we quantify it, and what we notice over the course of the study is that there's really this gradient where there's a high level of human disturbance here and a relatively low level of disturbance on the other end of the colony. So we're really starting to think of this as a gradient of urban disturbance or a gradient of fear. We're still figuring it out how to call it. And if we again take these data and we look at where the individuals that we tested are living, individuals that are living in these high disturbance areas tend to have a very short flight initiation distance. And this is consistent. These data are shown on the exact same scale, so that's important. And what you'll notice is that the other individuals on this low disturbance area are tending to be much more fearful than the individuals that are living in the areas that they often encounter humans. So we're starting to think about the influence that humans have on wildlife and using these squirrels as a way of testing this idea. And there are a number of other factors that are contributing to these decisions that these squirrels are making. So if we control for the starting distance of the walker, there's as the distance that we start, that actually influences their decisions. But in addition to this human disturbance, the distance that a squirrel may be to a burrow, so they'll go to these burrows for safety, that matters. They're much more relaxed with us approaching if they are close to a burrow. The incline of the habitat also matters. We can get closer when they are on an incline, as with the foraging behavior. However, we find one difference between this study and the study with the foraging behavior, in that if they are with another squirrel, they're actually more likely to flee at a shorter distance, or more likely to flee as we approach them, meaning that they would flee at a longer distance from us. So they're more fearful if they are with someone else. And we think that this is because there's this benefit of being with someone else, they're detecting the walker first, and that's giving rise to this conservative response of getting out of the way. Okay, so we're starting to think about these kinds of questions. And overall, we found that the foraging decisions that these squirrels make depend on social factors, onto genetic factors, their life stage, as well as the microhabitat in which they're in. And with the long-term data, we're actually able to, with a large sample size over multiple years, be able to think about context-dependent, risk-sensitive behavior. We're also finding consistent responses along disturbance gradient. And this will offer insights into the ways that humans are influencing wildlife as we visit them in these parks that are in part meant for them to be thriving. So what we're thinking about in future questions with this is another adult here. This is capital K. It's a rattlesnake if you haven't seen it yet. Capital K is one of the most bold individuals in our flight initiation distance test. She's also incredibly bold, and you're probably not even with this rattlesnake, and I'm taking this, trying not to shake, and so like, oh, capital K, you're doing okay. 5-year-old female, pups, etc. And what we're thinking about is the extent to which urban disturbance or contact with humans might actually make individuals less sensitive to natural predators. So to what extent does our influence on their behavior influence mortality and so forth? And capital K is doing just fine. She's still living her life out there, and I'm going to show you her in a social network pretty shortly here. Okay, so talked about foraging and risk-taking. To wrap this up, I'm returning to this theme of social networks and thinking about it here with California ground squirrels and the two distinct ecological contexts that they find themselves in. And basically, I've been thinking about more recently that many animals actually find themselves in two or more major ecological circumstances. So in nature, we often see animals, and even humans, we can think of ourselves, we're in closed spaces where we have social interactions. Birds might be in a nest, or these gelatos might be on a sleeping cliff. And then these same sets of individuals then move to a second ecological context, and in that context, interact with each other as well. And so what I'm interested in, again, is these social rules, and whether the rules that are governing behavior in this one ecological context is contributing and related to rules that are occurring in another context. And most of the work I've shown you so far has been in these really conspicuous, obvious, above ground scenarios. These squirrels are diurnal, they're easy to observe during the day. But they are semi-fossorial, and what that means is that they socialize, at least to the extent that we know, both above ground, such as sometimes they even climb trees, so, and then also below ground. And we really don't know much about their below ground social networks at all. And so this is the work, I have a grant with Andy C. at UC Davis, and we've been thinking about ways that the below ground architecture might influence the social networks that we see above ground. And this came out in a special issue focusing on how architecture may influence the emergence of social structure in relatively open spaces. And so you can see there's a lot of connectivity that's going on here, but up until now I've only focused on this aspect, what's been going on above ground. And introduce my other co-authors. So Chelsea Ortiz, again, she's now at Davis. This is a watermelon Wednesday. We don't have this every day, but a hot day at Briones, breaking open watermelon in the field. There's a squirrelmobile. Rihanna Hunter and Denise Gamboa and Julia Spencer all are undergraduates, so all undergraduate team here for this work. And basically, other than eating watermelons, we've been asking a lot of scientific questions. And the first question is, are the social network traits consistent within a particular context, but across time? So as these individuals are growing up, for example, or as adults are living from summer to summer, how do their social network connections vary across time, or are they actually consistent? And then second, to what extent does below ground connectivity in a social network predict what we see going on above ground? And so what we do for this work, as before, we live trap our squirrels. Lesko Munoz is here. We have some of these fecal samples for the court that I talked about briefly. Different squirrels that we mark, but importantly, we also give them a microchip or a pit tag, as you probably know, but a microchip would be what you have on a dog. You can scan them, and this pit tag allows us to have a unique identifier for each individual across their whole lifespan. And what we developed here is a new way of detecting their activity as they go in and out of these burrows. So there's this antenna, and it's situated at the entrance of this burrow. Every time a squirrel goes in or comes out, we have a timestamp. We know the identity of who was there. All of this information is then logged on a data logger that's powered by a rechargeable battery that we only need to change out every couple of weeks. So the heaviness of this equipment is on the ground, not on the squirrel. The squirrel just has to carry around this little tiny thing the size of a grain of rice, and we get lots and lots of data. It's actually being collected as we speak right now. From these data, we are able to combine what's going on below ground with traditional above ground observations. Here's Minnie with her binoculars watching squirrels, and we record in our notebook social interactions. And the above ground social interactions that we've been monitoring include, so for this work we're focusing on affiliative behavior, we also record aggression, but we wanted to know about things like, so these are greetings that squirrels engage in. They do a nose to cheek or nose to nose. They also are engaged in a number of different types of play. Here's a play pounce or even a play slap. And they also maintain social proximity. So to what extent are these networks of affiliative friendly behaviors related to who they are sharing space with below ground? And based on these interactions, we build up social networks, and I have, this is a toy social network with just 3 squirrels, A, B, and C. And what you can see here is that the circles represent juveniles and the squares represent adults. And the thickness of the line, or the tie between these 2 nodes in the network, reflects here the extent to which they're affiliating or exchanging these friendly behaviors with each other. So the red line represents these above ground social networks, and I filtered the network just so you can see some of the structure a little bit better, but the original analyses were done on all of the continuous data, which range from 0 to 1 for each squirrel in the network. These are above ground networks for these exact same sets of individuals. I then constructed networks based on their below ground behavioral interactions. And it looks like this. You can see the same individuals are here. The only difference is that we have the blue line is now reflecting shared occupancy of these burrow entrances. So on a given day, to what extent do they overlap in their below ground connectivity? A couple of notes on the social metrics that we used. We extracted 2 social network statistics. The first is the same type of measure that we used in the hyenas to look at the response to prey abundance across the network, standardized strength. And this simply reflects the direct ties, so how directly connected is A to B, B to C, and you can see A and C are not actually directly connected at all. So this tells us about how directly connected they are. And from looking at this toy set of networks, you can see that the highest strength is for squirrel B here. And that's interesting, but the real strength, sorry for the pun, of social networks is the looking at indirect connectivity. So not only are direct connections important, but indirect connectivity is also key. So you can see here in this toy network that squirrel B here is directly connected to A and C, but A and C are only indirectly connected through squirrel B. So this is very important when we think about how central animals are with respect to knowledge or information transfer, and also the transmission of diseases or parasites across social networks. So centrality indirectly connects individuals across the overall network. So we looked at both of these measures. Centrality is high for this one and not high for those other 2 more peripheral squirrels here. We looked at that in these two different social network contexts and for the same individuals across time. Importantly, as most of you experienced back in 2016, we were in a drought, and so the data that I will be showing you come from 2 years of the study. First summer in 2016, where the study area looked like this, and then 2017, where typically looks like this. And again, we wanted to know whether the social traits, both the direct strength as well as centrality, are consistent within a context. And what we did here is we focused on these 19 individuals within the social network to ask to what extent is their sociality consistent from the first year, 2016, to the second year, 2017. Okay? So we're focusing on individuals that are available in the network in these 2 consecutive years. To what extent are these social traits consistent across time? And what we found sort of a pattern, but not a statistically significant one, but a trend for strength in 2016 to correspond to the strength for that exact same individual in 2017. We looked at centrality, again, sort of a positive relationship, a trend, but not statistically significant. And this is in the above ground context. If we ask the exact same question, but now we're working in blue, these are below ground contexts, to what extent are social traits consistent below ground in these social networks? And here we actually find that the strength of individuals in 1 year, that same individual has a high strength in 1 year, has a high strength in the next year. If we look at how central they are, the indirect measure of connectivity, centrality is also significantly consistent across these 2 different years. So this suggests that below ground individuals have consistent social network traits across time, even though we're going from 1 year to the next. So overall, social personalities, mainly below ground, so they're social personalities because they're fairly consistent across time. Some individuals are highly social, others are loners, such as this one. And this consistency is mainly below ground. But to what extent are these interactions below ground predicting what we see above ground in terms of social connectivity? Can what's going on in one ecological circumstance promote or give rise to what's going on in another social situation? So here, we looked at the overall structure of the network from 2016. This is below ground, and we predicted that the architecture of these interactions below ground is constraining where individuals and who they're interacting with above ground. So we predicted this below ground network in 2016 would predict the above ground social network. We used a multiple regression that is specific for social networks, this MQuAP basically, which controls for non-independence in the data. We control for similarity in age and sex categories, and we find a significant relationship between social network connectivity below ground and that's going on above ground. So this is at an overall level of connectivity, and again we see the exact same pattern in 2017, suggesting that the individual connectivity below ground is consistent with what's going on above ground. If we focus on this in a little bit more of a specific way again, and focus on particular individuals, and ask to what extent is their strength below ground predicting their strength in the social networks above ground, we also see a significant relationship between the two. Here I've simply ranked the below ground strength from 0 to 60. There are 60 individuals here. I've ranked them because the scale of the social network connectivity differs below ground compared to above ground. And I just put them into 4 different bins to be able to see them. But what we find when we do the permutation tests is a consistent relationship where what's going on below ground is predicting what's going on above ground. This is during the drought year, so still resilient to that. And we have an even stronger pattern the following year of what's going on below ground with respect to how strong these social network connections are with what's going on above ground. If we look again at centrality, how central these connections are, and use the same methods, we fail to find a consistent pattern in centrality below ground and above ground in that drought year. But if we go to the next year, go back to the field, in a typical year, we do find a significant pattern of what's going on below ground in terms of how central individuals are in the network and how central they are in these relatively free and open social environments. So overall, we find that these social traits are fairly consistent within contexts across time and also between contexts. And what we're starting to think about is, I always like to talk about what we're thinking about next. And we're trying to think about ways that this continuum of how sociable these individuals are might influence things like disease transmission. And squirrels, as probably many of you know, they can carry many fleas, and these fleas have even been shown to carry plague. Now, the squirrels that we work on are healthy, but they still offer a very interesting model for thinking about transmission and social transmission of diseases and parasites across time and space. So we're starting to think about that in these two different ecological situations. And Bob Lane, in entomology here emeritus, wasn't able to make it today, but this is work in collaboration with him. Many students have been contributing to this work, and I'll just show you one year of data for individual squirrels ranked by their flea count. If we if we comb them on the first time we've captured them of a week, we comb them 5 times, we count up the number of fleas. They're remarkably consistent in how many fleas they have, and this is after controlling for day of the year, their age and sex category, all of these things. Some individuals consistently have a large number of fleas and others have very few. So we're starting to think about the extent to which social network connectivity may actually give rise to these ectoparasite loads. And with that, I'd like to thank all of the members of Team Squirrel. There are many that have been helping out with this part of the work. I would also like to thank my funding sources and you for listening and take questions if you have them.
Hit the lights in the back corner there. I know some of you may have to run for classes, but just for a few minutes, let people get out. Boss is already out.
Well, that was a teaser at the end. So are the central individuals the ones with the most fleas?
Yeah, so we're still working on that. That's why, so we clearly know that if I pick up a squirrel, I know who they are based on their mark. When they come to me at the trap station, I'm like, oh my gosh, umbrella with your 92 fleas. So we know that. I have an intuition that those same individuals are more directly connected in the social network, but we're still starting to tease that apart. There's, I talked a little bit about the social dynamics with the hyenas, and for the looking at these ectoparasites, they have a temporal dynamic. So we actually need to break up the networks, not in the same way those are just binned over the whole summer, but we need to do time slices because there's 2 species of fleas that we're dealing with. Each one has a seasonal peak. So the short answer is I don't know yet, but I very much suspect that the individuals that have the most babies, that share a lot of burrow use below ground, are probably the ones that have the most fleas. And this probably relates to above ground behavior, but I suspect that the below ground connectivity is the most important driver there, because the fleas will hop off and hop back on.
Other questions?
Yeah.
So we certainly have enough interesting questions to follow up with. This question about the fleas and also makes me think, do you have any predictions about how that might affect the number of fleas or the state of the animal or the risk-taking behavior?
Absolutely. So we're thinking a lot about that, and I was just talking with Chelsea planning out her dissertation yesterday. We are thinking a lot about these social factors, the physiological state, and actually collaborating with Eileen thinking about glucocorticoid metabolites. So in the relationship of that and immunocompetence and ectoparasite load, so I think they all are related, and the degree of human disturbance influences their stress response. So their baseline stress response is higher, we hypothesize, at least at the colony level we know, but we hypothesize along this gradient as well, that there's a difference in their circulating hormones that is modulating their risk-taking behavior. We're looking at links between that and also alarm calling. Their alarm calling is the extent to which they do that when we approach them is very individually specific. and the acoustic properties of that is also individually specific. So identity, identity, identity. It's really quite interesting, these little squirrels that, you know, kind of look the same to us. They're not very sexually dimorphic, they're a little bit, but for the most part you wouldn't necessarily know. But the long-term data are actually suggesting there are these really interesting individual differences, and in all these models it's pretty much falling out as one of the main contributing factors. Yeah.
Bill, yeah.
Yeah,
I'm sorry.
I was really interested at the drama of the individual differences predominating. That was so dramatic. What are you thinking about? What's the source of those individual differences? Is there any developmental role in that, or just what have your thoughts been on that subject?
Yeah, so for the individual differences, I assume that is with respect to risk sensitivity. That figure thinking about how close we can get to them, and some individuals are flying away pretty quickly, others we can get very close to. We're thinking about how that maps onto this gradient of fear across the landscape, so where they're living. We're thinking about questions of so they have these burrow systems that they're very much faithful to. We will ask whether individuals are choosing to be in the areas of high disturbance, or if high disturbance over evolutionary time might actually be favoring individuals to behave differently. So we're building up a pedigree to look at across different generations and start to ask questions that way. Yeah, so or is it maybe just something learned? I see humans a lot and I habituate to them. It might be something like that. For other tests that we've done with the pups where we've given them different predator treatments, the juveniles don't seem to understand those threats very well compared to the adults, but there might be some learning that's going on there.
Quasi, you had your thing up.
Yeah, so for centrality between below ground and above ground, in that drought year when we didn't observe much of an effect, do you have any ideas as to why you didn't really see any relationship, even though the effects seemed to carry over into the non-drought year?
Yeah, so we had lots of strange things were happening in that year, and it's just a one-off, right? So it'd be really interesting to look at it as we start to accumulate more data. But it suggests that when there's a drought, that's a perturbation to the network, and that actually breaks up this consistency that you might otherwise have. And so it might be something to do with literally just the water flooding burrow systems, changing the architecture of these burrows. It might also be high levels of predation. Hard to say. Actually, coyotes were not doing well with the drought, so there might be lower predation. So lots of different factors. We're collecting information about all of them, and I think with more years we can start to get a better picture of that.
Yeah, there were a couple more. Is it Eric? Yeah.
Is there any, what's the degree of, I guess, food competition between the squirrels? Is there any dominance systems in place there that might have something to do with the individual variation in their sort of sociality?
Yeah, so we haven't looked at dominance hierarchies. I mentioned I was working with marmots for a postdoc at UCLA, and there we actually constructed dominance hierarchies that were consistent with individuals as juveniles in their play bouts. Those outcomes predicted what was going on in adulthood. We haven't looked at that yet in the California ground squirrels. We don't see that many agonistic interactions, and that would be what we would need to build these on. We definitely see structure with respect to the play behavior and who's initiating play more often. The males are typically doing that. And who's winning play, males are doing that as well. So that would suspect that there's maybe some maintenance there, and that's giving rise to these sex differences. But to the extent that there's a hyena dominance hierarchy, probably not. Yeah.
Maybe one more. Alexander, yeah.
So that was a really awesome talk on squirrels and hyenas, but I did notice a lot of videos of snakes also. So I guess my question is, have you thought of pit tagging snakes and seeing how, I've seen a lot of rattlesnakes enter squirrel burrow systems in Briones, and could that potentially impact their social interactions in any way? Or?
Absolutely. So sometimes we do have snakes that reside in a particular squirrel burrow, and we do note that. And there probably are opportunistic, if a snake eats a squirrel, they may have a pit tag in them until that leaves them. And Rulon Clark at San Diego State University does put pit tags at times in rattlesnakes. That's not something that I have taken on, but they do collect the snakes and they'll put a pit tag in when they're anesthetized and put them back out. Absolutely, predation is shaping pretty much every aspect of the squirrels' lives, and so it'll be really interesting to look at these questions in more detail from both perspectives.
So Jenna, fill in, I think you probably have a couple herpetologists who are willing to volunteer to do that if you really wanted to do
Okay.
it.
Perfect. I'm probably out of.
Maybe one last question, anyone have one before we let Jen get a break? Yes.
It sounded from what you said that you may have looked at this, but I wonder how this flight distance is related to age and also to sex. But I'm wondering about the learning. Presumably the humans are in fact harmless here, and whether they learn that and know more as they get older and therefore are less sensitive.
Yes, so as I said, this work is still kind of, we're building up the data set, but preliminarily what it looks like is that adult females will allow us to get closest to them. So that's interesting because that suggests that maybe they're habituated to us, particularly these females that have been around for a long time. They live in that area, they're natal to it, they're remaining in this, they understand, they've seen us for many years now, because we just started doing that this last summer. So it's suggestive that these older individuals are more habituated to us, whereas males, even if they're adults, are less likely to allow us to get closer to them. So and they may have immigrated from elsewhere.
And the one outlier on the less disturbed area, could that possibly be a migrant?
Could be. Could be. Yeah, so it's a really interesting data set and really easy to get these type of data, and the squirrels are just remarkably consistent, so we can start teasing apart lots of different factors from them. Thanks for asking. Yeah.
Cool. Well, let's thank Jen. Some of you have signed up to stick